Hello everyone, I'm Leo Zhang Guoliu, an interdisciplinary researcher in visual neuroscience and computer vision. In this video, we will explore the analog-to-digital conversion mechanism in the human retina. Through this video, I will attempt to answer the following 4 questions. First, how does the human retina convert creative potential signals from photoreceptor layer into action potential signals at ganglion cell layer? Second, what functional role do retinal cells play in the retina, and what is the working principle behind their function? Third, exactly how many colors can human eye distinguish? Fourth, what are the encoding principles of biological nerve systems based on spike chains? Now, let's get to the main topic of this video. The organizational structure of the human retina is shown in the figure. It primarily consists of 3 layers of cells: the photoreceptor layer, the bipolar cell layer, and the ganglion cell layer. When light enters the eye, it first passes through the upper 2 layers of cells to reach the photoreceptor layer, where it is converted into neuroelectrical signals by photoreceptors through photochemical reactions. These neuroelectrical signals are then processed by bipolar cells and ganglion cells before being transmitted to the lateral jet nuclear nucleus and the visual cortex of the brain via the optic nerve bundle, which is composed of the axons of all ganglion cells. Therefore, the direction of signal transmission in this diagram poses from bottom to top. Upon receiving light signals, photoreceptors convert them into memory potential signals ranging from -40 mV to -70 mV through photochemical reactions. If we assign light intensity values on a scale of 0 to 255, we can simplistically describe the photoreceptor's function as encoding these 0 to 255 light intensity values using memory potentials between -40 mV and -70 mV. For example, a memory potential of -61.0 mV might encode a light intensity of 178.5 units. A memory potential of -61.1 mV might encode a light intensity of 179 units, and so on. Since these memory potential signals can theoretically assume infinite possible values within the -40 mV to -70 mV range, with each distinct value corresponding to a different light intensity level, they are termed graded potential signals. Moreover, as these graded signals can represent continuously varying physical amplitudes through continuous voltage changes, they are also regarded as a form of analog signal. However, at the ganglion cell layer, the signal form has changed. Ganglion cells employ action potentials to encode and transmit information. When a ganglion cell is not activated, its memory potential remains at the resting level. Once the activation threshold is reached, the memory potential undergoes millisecond-level rapid depolarization, followed by immediate repolarization to the resting state. This positive electrical activity is termed an action potential. Action potentials encode all information using just 2 states, resting and activated, which bears remarkable similarity to how the digital world encodes all information using just zeros and ones. Consequently, the scientific community generally considers action potential signals to be a form of digital signal. The human retina encodes signals in analog form at photoreceptor layer and in digital form at ganglion cell layer. This indicates that between these 2 cellular layers, the signal undergoes a conversion from analog form to digital form. In the fields of electronic engineering and communication engineering, this signal transformation has a specific technical designation: analog to digital conversion, abbreviated as ASD. In other words, within the human retina, there exists an analog to digital conversion mechanism. We may refer to it as the biological ADC. Our subsequent research objective is to decipher this biological ADC mechanism within the human retina. We can accomplish this research objective through 2 sequential tasks. As evident from the diagram, the retinal signal transduction pathway involves only a single synaptic mapping through the bipolar cell layer between the analog signal output from photoreceptor layer and the digital signal generation at ganglion cell layer. This indicates that the retinal analog to digital conversion is achieved in a single processing step. Therefore, our first research task is to identify an algorithmic solution capable of performing one-step analog to digital conversion. Upon identifying such an algorithm, we then need to complete the second task: obtaining biological evidence demonstrating that the retina employs this specific algorithm, and further elucidating the precise biochemical mechanisms through which the retina implements this transformation. Let's first address the initial task, identifying an algorithmic solution capable of performing one-step analog-to-digital conversion. We analyze binary encoding principles by examining a 5-bit binary code. A 5-bit binary code can represent 2 to the power of 5, that's 32 discrete quantization levels, covering a numerical range from 0 to 31. First, we enumerate all 32 numerical values. Next, we encode these 32 values using 5-bit binary code. Value 0 is encoded as 5 zeros. Value 1 is encoded as 4 zeros and a 1. Following the binary carry rule, where 1 is carried when reaching 2, value 2 is encoded as 00010. Then, value 3 is encoded as 00011. And after processing, we can determine value 4 is encoded as 00100. And so forth until we obtain all 32 binary codes. We then mark all 1 values in this binary array in blue, and separate each bit for individual observation, yielding the result shown in the figure. This figure shows that at every bit position in binary code, the distributions of 0 values and 1 values exhibit perfect regularity. We define any complete continuous sequence of zeros at a bit position as a 0 interval, and of 1s as a 1 interval. Here's what stands out. Firstly, for every bit position, its 0 intervals and 1 intervals are of equal length. Secondly, they alternate strictly in a 0-1-0-1 sequence without exception. At any bit position, we define a pair of 1-0 interval and its immediately following 1 interval as a 0-1 circle. In conventional numbering, bit positions are ordered from least significant bit as bit 1, bit 2, bit 3, etc. For analytical convenience, we adopt reverse ordering from most significant bit, designating them as bit top 1, bit top 2, bit top 3, and so forth. The figure clearly shows that from bit top 1 to top 5, the counts of the 0-1 circles are precisely 1, 2, 4, 8, and 16. Each subsequent bit position has exactly twice the number of 0-1 circles as the previous one. Therefore, we can summarize the following law. Under binary code rules, all bit positions top I precisely partition the value range into exactly 2 to the power of I-1 equal 0-1 circles, as shown in these equations. I name this law the 0-1 circle law of binary code. By applying the 0-1 circle law, we can achieve highly efficient conversion for analog signals to binary digital signals. In electronic engineering, the device that converts analog signals to digital signals is called an analog-to-digital converter, short for ADC. We take the design of a 4-bit resolution ADC as an example. A 4-bit ADC can distinguish and encode 2 to the power of 4, that's 16 discrete quantization levels, covering a numerical range from 0 to 15. We first list all these 16 levels. Next, we configure 4 parallel subcircuits that divide the numerical range into 1 0-1 circle, 2 0-1 circles, 4 0-1 circles, 8 0-1 circles. Assume the sensor first converts light intensity values ranging from 0 to 15 into corresponding voltage values from 0 volts to 5 volts. Thus, the value range of each subcircuit becomes 0 volts to 5 volts. Once the simple configuration is completed, the ADC is fully operational for analog to digital conversion. For example, suppose we receive a light signal with amplitude 13. The sensor first converts it to a 4.3 volts voltage value, which is then fed in parallel to all subcircuits. Each subcircuit then determines its output based on whether the 4.3 volts falls within its 0 interval or 1 interval. In this case, the 4.3 volts is located in the 1 interval, 0 interval, and 1 interval of the 4 subcircuits respectively. Therefore, the output values are 1, 1, 0, 1. The resulting digital sequence 1101 is exactly the binary representation of the analog value 13. The analog to digital conversion is completed. Since this method utilizes the 0-1 circle law of binary code to achieve rapid analog to digital conversion, I named it the 0-1 circle method, and refer to the ADCs employing the 0-1 circle method as 0-1 circle ADCs. To be more specific, We can implement this design by equipping of the four subcircuits with the dye component as illustrated. The current conduction path of the component is input terminal, pointer base, pointer tip, circular ring, output terminal. The pointer is designed to rotate proportionally to the input voltage, completing a full 360-degree rotation at 5 volts. Furthermore, we partition the circular ring of each component according to its bit position into 1Z1 circle, 2Z1 circles, 4Z1 circles, 8Z1 circles. With this configuration, the circuit acquires analog to digital conversion functionality. For example, when receiving a light signal with amplitude 13, the sensor generates a 4.3 volts voltage. This voltage corresponds to a 311-degree rotation, causing all four pointers to rotate to 311 degrees. At this angle, the pointer tips on the four subcircuits lie in the 1 interval, 1 interval, 0 interval, and 1 interval. Thus, the outputs are 1, 1, 0, 1. The resulting digital sequence 1101 is precisely the binary representation of the analog value 13, completing the analog to digital conversion. Since this component implements Z1 circle configuration to test the division of the circular ring, I hereby named it the sector type Z1 circle component, and ADCs incorporating such components are accordingly named sector type Z1 circle ADCs. Observing the sector type Z1 circle ADC, we recognize that by applying the Z1 circle law of binary code, the analog to digital conversion requires just one operational step. Each subcircuit simply needs to determine whether the analog signal's amplitude falls within its 0 interval or 1 interval, and outputs 0 or 1 value accordingly, thereby completing the entire analog to digital conversion. As previously discussed, the human retina converts analog signals from photoreceptor layer to digital signals in ganglion cell layer to just a single mapping layer of bipolar cells. Using our newly discovered Z1 circle method, we can now simulate this biological functionality. For example, suppose the photoreceptor emits an analog signal with amplitude 13. How does the human retina convert it into digital signal 1101 at the ganglion cell layer to a single mapping in the bipolar cell layer? A feasible biological implementation would be each photoreceptor distributes the analog signal to 4 bipolar cells. We configure these 4 bipolar cells with 1, 2, 4, and 8Z1 circles respectively, with each connecting to one ganglion cell. Based on this configuration, when receiving the analog signal, each bipolar cell simply determines whether to activate its downstream ganglion cell based on whether the signal value falls within the 0 interval or 1 interval, thereby directly yielding the 1101 digital signal at the ganglion cell layer. Remarkably, this conversion process concludes in just one operational step. Moreover, we observe that the 4 independent subcircuits in the diagram can largely match the well-ordered histological architecture of the bipolar cell layer. This suggests that the human retina may indeed utilize the Z1 circle method to accomplish rapid analog to digital conversion from photoreceptor layer to ganglion cell layer. However, to substantiate this hypothesis, mere theoretical feasibility and structural similarity are insufficient. We require more definitive biological evidence. Fortunately, such biological evidence is readily available to us. This set of wavelength sensitivity curves of photoreceptors is a staple figure in biology textbooks, and I believe everyone is familiar with it. It characterizes the wavelength-dependent sensitivity variations across the 4 photoreceptor types in human retina. The graph on the right shows the spectral response profiles of horizontal cells. It displays the stimulus-evoked firing patterns of horizontal cells in cup retina when stimulated sequentially with monochromatic light from 400 nm to 700 nm. Let's first review the basic information about horizontal cells. Horizontal cells are inhibitory neurons functioning in the outer plexiform layer of the retina, the synaptic layer where the photoreceptor layer connects with the bipolar cell layer. They modulate neurochemical signal transmission between these two layers. This figure illustrates that researchers have identified 3 types of horizontal cells in cup retina, each exhibiting distinct spectral response profile. These profiles are classified as L-type, the luminosity type, C-biphasic type, the chromatic biphasic type, C-triphasic type, the chromatic triphasic type. As shown in the figure, although the three types of horizontal cells display different response patterns, they share a common feature. All exhibit alternating periods of inhibition and excitation along the wavelength spectrum. Now, by analyzing these wavelength sensitivity curves of photoreceptors and these spectral response profiles of horizontal cells, we can derive the following two conclusions. First, the biological retina indeed employs the J1 circle method to achieve rapid analog to digital conversion from photoreceptor layer to ganglion cell layer. Second, horizontal cells are the key retinal cells responsible for analog to digital conversion in biological vision systems. Below is my derivation process. Assume the analog to digital conversion in the heme retina converts the creative potentials of long wavelength sensitive cone cells, which we refer to as red cone cells, into AB binary code. This means the heme retina divides the response values of red cone cells into 2 to the power of A, which is 256 quantization levels. These 256 levels correspond to a numerical range of 0 to 255. We call the response values of red cone cells R values, express their numerical range as 0R to 255R. Stimulate the red cone cells sequentially with saturated monochromatic light from 420 nm wavelengths to 705 nm wavelengths. For example, first use 420 nm wavelengths saturated monochromatic light, then switch to 425 nm, 430 nm, and so on, up to 705 nm. During this process, we can observe that the R values first gradually increase from 0R to 255R, then gradually decrease from 255R back to 0R. Let's assume the heme retina uses the J1 circle method to achieve analog to digital conversion. Then, these 256 R values from 0R to 255R need to be jointly encoded by A bipolar cells. Ordered from most significant to least significant digit, we designate them as I bipolar cell, R7 bipolar cell, and so on down to R1 bipolar cell. And their J1 circle distributions are as shown in the figure. It can be seen that within the numerical range of 0R to 255R, the I bipolar cell has 1 zero interval, the R7 bipolar cell has 2, the R6 bipolar cell has 4, the R5 bipolar cell has 8, and so on. Let's remember the fact that the R5 bipolar cell has 8 zero intervals within the 0R to 255R range. Now, when stimulated red cone cells with saturated monochromatic light from 420 nm to 705 nm, the spectral response curve is shown in the figure. Then, what would the spectral response profile of R5 bipolar cells look like under the same stimulation conditions? In the 420 nm to 575 nm range, the R value undergoes a process of climbing from 0R to 255R. As mentioned earlier, the R5 bipolar cell has 8 zero intervals within the 0R to 255R numerical range. Therefore, its response profiles in this range will be approximately as shown in the figure. In the 575 nm to 705 nm range, the R value undergoes a process of declining from 255R back to 0R, which similarly covers the complete numerical range from 0R to 255R. Therefore, the R5 bipolar cell will also have 8 zero intervals within this range, as shown in the figure. Thus, across the 420 nm to 705 nm wavelength range, the R5 bipolar cell undergoes a total of 16 zero intervals, with its complete spectral response profile displayed in this figure. So, what neural configuration should the retina employ to enable R5 bipolar cells to exhibit the response pattern shown in the figure? We have two alternative strategies. The first option is to configure photoreceptor cells to refrain from releasing neurotransmitters to bipolar cells during all zero intervals. The second option is to not interfere with neurotransmitter release from photoreceptor cells, but instead to simultaneously apply equal magnitude counterpotential to bipolar cells during their zero intervals. This will cancel out the activation potentials generated in bipolar cells, thereby maintaining them in a resting state overall. If the retina adopts the second strategy, then for the 16 zero intervals of R5 bipolar cells, the retina needs to apply counterpotentials of equal magnitude but opposite polarity, as demonstrated here, to R5 bipolar cells through some mechanism. If this is indeed the case, we should be able to observe the following two histological and cell biological phenomena in the retina. First, in the synaptic connections between photoreceptor cells and bipolar cells, there should exist a type of inhibitory neuron. This is because both photoreceptor cells and bipolar cells themselves are excitatory neurons and cannot generate inhibitory potentials. Therefore, the retina needs to incorporate a type of inhibitory neuron between photoreceptor cells and bipolar cells to provide the necessary counterpotentials. Second, these inhibitory neurons should apply counterpotentials during all intervals of bipolar cells. Therefore, we should be able to observe these neurons exhibiting a counterpotential profile approximately as demonstrated here. Examining the organizational structure of the human retina, we observe that at the synaptic connections between photoreceptor cells and bipolar cells, to be exact, in the outer plastiform layer, there indeed exists an inhibitory neuron, the horizontal cell. Simultaneously, we can observe that the L-type response profiles of horizontal cells shows remarkable similarity to our hypothesized counterpotential profile. The defining characteristic of this counterpotential profile is its alternating patterns of resting activity states across the wavelength spectrum. This core feature is present not only in L-type horizontal cells, but invariably in both C-type biphasic horizontal cells and C-type triphasic horizontal cells as well. These alternating patterns of resting activity states essentially represent the Z1 circle response. The presence of the Z1 circle response characteristic in horizontal cells strongly suggests that biological retina most likely employ the Z1 circle law in their functioning. Based on the above analysis of phenomena, I've drawn the following conclusions. First, the biological retina indeed employs the Z1 circle method to achieve rapid analog to digital conversion from photoreceptor layer to ganglion cell layer. Second, the Z1 circle response pattern of bipolar cells relies on the counterpotential regulation provided by horizontal cells. Therefore, horizontal cells serve as the key retinal cells enabling the retina's analog to digital conversion function. Regarding how horizontal cells regulate the Z1 circle response pattern of bipolar cells, I have considered possible biochemical reaction pathway. I must clarify in evidence that the following content represents only my personal hypotheses and may not correspond to factual mechanisms. Please exercise careful discernment when considering this information. It is known that the regulatory site of horizontal cells are located at the synaptic connections between photoreceptor cells and bipolar cells. Therefore, we incorporate the synaptic terminals of horizontal cells at these corresponding positions. When horizontal cells do not participate in regulation, the normal signal transmission proceeds as follows. After receiving light stimulation, photoreceptors generate memory potentials with corresponding amplitudes. These memory potentials propagate to the presynaptic membranes of photoreceptors, triggering neurotransmitter release from their presynaptic terminals. The chemical neurotransmitters then transmit to the postsynaptic membranes of bipolar cells. Receptors on these membranes receive these neurotransmitters, initiating a series of biochemical reactions that generate activation potentials here corresponding to the neurotransmitter concentration. This is the unregulated signal transmission process. We refer to the biochemical reaction process responsible for triggering activation potentials as the activation reaction chain. Then, how do horizontal cells participate in this reaction process and exert their influence? My hypothesis is that the synaptic terminals of horizontal cells have the same receptor distribution as the postsynaptic membranes of bipolar cells. Therefore, the neurotransmitters from photoreceptors are received by both bipolar cells and horizontal cells simultaneously. After receiving neurotransmitters, horizontal cells generate inhibitory potentials through a series of biochemical reactions. These potentials have equal amplitude but opposite polarity to the activation potentials of bipolar cells, and are what we call counterpotentials. Unidirectional ion channels exist between the synaptic terminals of horizontal cells and the postsynaptic membranes of bipolar cells. The counterpotentials from horizontal cells rapidly propagate to bipolar cell postsynaptic membranes through these ion channels. Since electrical conduction is much faster than neurotransmitter transmission, and the spatial distance between horizontal cell terminal and bipolar cell postsynaptic membrane is extremely short, the conduction time of counterpotentials is practically negligible. This enables horizontal cell counterpotentials to reach bipolar cell postsynaptic membranes before the activation potentials depart. successfully intercepting the activation potentials and preventing their downstream propagation, thereby maintaining bipolar cells in the resting state. Now, the next question is, how do horizontal cells determine the timing of regulation? In other words, how do horizontal cells discern when to apply counterpotentials to bipolar cells and when to refrain from doing so? We refer to the biochemical reaction process that generates counterpotentials in the synaptic terminals of horizontal cells as the regulatory reaction chain. My personal hypothesis is that within this regulatory reaction chain, there should exist a biomacromolecule functionally analogous to a geometrical component. We tentatively designate it as the switchosome. The switchosome is one link in the regulatory reaction chain and rotates to corresponding angles based on neurotransmitter concentration. When the switchosome rotates to an active sector, it assumes an active conformation. At this point, the horizontal cell's regulatory reaction chain pulses unimpeded, triggering counterpotentials that prevent the propagation of bipolar cell activation potentials downstream, thereby maintaining the bipolar cell in a resting state. And when the switchosome rotates to an inactive sector, it transitions to an inactive conformation. At this point, the regulatory reaction chain in the horizontal cell terminal is arrested, rendering it unable to trigger counterpotentials. Consequently, the activation potential of the bipolar cell pulses unimpeded and can propagate downward, thereby activating the bipolar cell. The switchosomes adopt a 1-0 circle configuration, meaning their rotational angle ranges are divided into multiple 1-0 circles. Specifically, in horizontal cells regulating RA bipolar cells, the switchosomes' rotational ranges are divided into one 1-0 circle, as shown in the figure. We designate this kind of switchosome as the HA switchosome, and horizontal cells containing HA switchosomes as HA horizontal cells. Correspondingly, horizontal cells regulating R7 bipolar cells are termed H7 horizontal cells, containing H7 switchosomes in which the rotational ranges are divided into two 1-0 circles. By extension, with H6 switchosomes, the rotational ranges are divided into four 1-0 circles, and so forth. When a photoreceptor receives light stimulation, it releases neurotransmitters at identical concentrations to its A-graded types of bipolar cells downstream. Consequently, all switchosomes involved in regulating these bipolar cells rotate to identical angles, as shown in the figure. However, due to their distinct 1-0 circle configurations, the activation states differ at the same rotational angle. For example, in the illustrated case where switchosomes rotate to 252 degrees, H8 and H6 switchosomes assume inactive conformations, and H7 switchosome assumes an active conformation. Taking the R value of 179R as an example, I will demonstrate my hypothesized biochemical process for retinal analog to digital conversion. First, the retinal cell releases neurotransmitters at identical concentrations to all bipolar cells responsible for encoding different bits. These neurotransmitters are simultaneously received by synaptic terminals of A grades of horizontal cells, causing switchosomes to rotate to 252 degrees. At the synaptic connection site of RA bipolar cell, the HA switchosome assumes an inactive conformation after rotating to 252 degrees. The regulatory reaction chain in the horizontal cell terminal is arrested. The counterpotential is not triggered. RA bipolar cell activates normally without inhibition. Downstream ganglion cell is activated. One action potential is generated, corresponding to encoded value 1. At the synaptic connection site of R7 bipolar cell, the H7 switchosome assumes an active conformation. The regulatory reaction chain pulses unimpeded. The counterpotential is triggered. R7 bipolar cell is inactive due to this inhibition. Downstream ganglion cell is not activated, corresponding to encoded value 0. The same applies to the rest of the synaptic connection sites. Here, we propose a new concept termed the on-off array, which refers to the combination of on-off states at the same time point of a neuron group within the same layer. Based on this definition, the final on-off array of the above 8 ganglion cells appears as shown in the figure, and its corresponding digital sequence is 10110011, which is precisely the binary code for 179. The analog to digital conversion is thus completed. From this, we have outlined the potential biochemical process by which the human retina might achieve analog to digital conversion. While the specific details may not be entirely accurate, the intended function is successfully achieved. However, there remains an obvious issue with this hypothesized mechanism of biological ADC. If the conversion process indeed operates as illustrated, it will require each photoreceptor to connect to A bipolar cells and A ganglion cells, implying a numerical ratio of 1 to A among these three cell types. Yet this is not the case in reality. This table demonstrates the actual numbers of excitatory neurons in the human retina. Specifically, there are approximately 120 million photoreceptors, 35 million bipolar cells, for only 1.2 million ganglion cells. The numerical ratio among these three cell types is approximately 100 to 30 to 1. With so few ganglion cells, it is fundamentally impossible to support the analog to digital conversion demands from hundreds of millions of photoreceptors. How should this be explained? Regarding this issue, my personal analysis leads to the following conclusion. Ganglion cells encode not the receptive field of individual photoreceptors, but rather the receptive field of single pixels, where each pixel's receptive field is composed of multiple photoreceptor receptive fields, as shown in the figure. I term this illustrated structure the vision neural model. The vision neural is a concept I propose, referring to a retinal block unit composed of all cells encoding visual information from a single spatial point. It represents the retina's smallest functional imaging unit. Thus, the human retina can be conceptualized as a pixel matrix formed by numerous contiguous vision neurons. For example, 16 vision neurons can be assembled to form a 4x4 pixel matrix retina, as shown in the figure. Note that in the actual human retina, the photoreceptor layer is positioned at the basal-most position relative to the light path direction, as illustrated. Here, for demonstrating purpose, the vision neural model is vertically inverted, placing the photoreceptor layer on top. Please take slight note of this distinction. When aligning with the actual retinal structure, the vision neural model should be orientation reversed. Now, I'll use this vision neural model as a demonstration tool to explain my personal hypothesis about how the retina accomplishes analog to digital conversion. The vision neural model includes three color channels: red, green, and blue. The numerical value of each color channel is encoded by 3 ganglion cells, and each ganglion cell receives input from 9 photoreceptor cells and 9 bipolar cells. Taking the red channel as an example, the R value is jointly encoded by R3, R2, and R1 ganglion cells. The R3 ganglion cell receives input from uniformly distributed 9 photoreceptor cells and 9 bipolar cells of the same type within the vision neuron's receptive field, and requires more than 50% upstream bipolar cell activation to fire. The same applies to the R2 ganglion cell and the R1 ganglion cell. After extending this arrangement to all ganglion cells, we obtain the neural wiring of the vision neuron as shown in the figure. The purpose of this setting is to filter out light stimuli with illumination area below the threshold, preventing unnecessary consumption of the brain's computational resources. As illustrated, the red beam's illumination covers less than 1/3 of a single pixel area. Such mini stimuli should be excluded from visual processing. Now, the following question is, how should the vision neuron be configured to ensure that the 9 bipolar cells providing input to the same ganglion cell share identical encoding properties? To begin with, we must clarify that identical encoding properties entails 2 requirements. First, these bipolar cells must encode the same color channel. Second, they must encode the same digit. For example, all bipolar cells providing input to the R2 ganglion cell must encode the red channel and digit 2. To make 9 bipolar cells encode the same color channel is straightforward. We simply have them receive input from the same photoreceptor type. For instance, by having all 9 bipolar cells in the diagram receive input from red cone cells, they will automatically encode the same color channel. Then, how can we make 9 bipolar cells encode the same digit? For example, how can we make the 9 bipolar cells in the diagram all encode digit 2? The method is also straightforward. We just need to assign one H2 horizontal cell to the vision neuron and have all 9 bipolar cells be regulated by still one circle modulation, as shown in the figure. The same principle applies to G2 bipolar cells of the green channel and B2 bipolar cells of the blue channel. We simply need to have them receive input from the green cone cells and blue cone cells respectively, while also being regulated by the H2 horizontal cell, thus completing the configuration. Here we can note that, since the J1 cycle modulation by horizontal cells relates only to the neurotransmitter concentration of photoreceptor cells and is independent of photoreceptor cell types, all bipolar cells encoding digit 2, regardless of which color channel they belong to, can be regulated by the same H2 horizontal cell. The neural wirings encoding digit 1 and digit 3 follow the same rule as digit 2. When we incorporate them into the model, we obtain the complete vision neural model as shown in the figure. Now, let's demonstrate the workflow of the vision neural in detail. Each color channel in the vision neural model is encoded by 3 ganglion cells. Therefore, its data resolution is 3 bits, capable of distinguishing 2 to the power of 3, that's 8 quantization levels, with a numerical range from 0 to 7. Assume a light stimulus is currently projected on the vision neuron's receptive field, triggering red, green, and blue cone cells to produce response values of 6R, 2G, and 3B respectively. Then, for the red channel, 6R falls within R3 and R2's 1 intervals, R1's 0 interval. Therefore, R3 and R2 bipolar cells are activated, while R1 bipolar cells remain at rest. For the green channel, 2G falls within G2's 1 interval, G3 and G1's 0 intervals. Therefore, G2 bipolar cells are activated, while G3 and G1 bipolar cells remain at rest. For the blue channel, 3B falls within B2 and B1's 1 intervals, B3's 0 interval. Therefore, B2 and B1 bipolar cells are activated, while B3 bipolar cells remain at rest. Thus, after receiving the current light stimulus, the bipolar cells and ganglion cells will exhibit the on-off state changes as shown in the figure. At this point, the on-off array formed by the 9 ganglion cell states corresponds to the digital sequence 110, 010, 011. This digital sequence signal, after long-distance transmission through the optic nerve bundle and optic radiation fibers, arrives intact at the visual cortex. The visual cortex then decodes this digital sequence, thereby accurately determining that the color value of the light stimulus is 6R, 2G, 3B. The above represents my personal hypothesis regarding the signal processing workflow of the human retina. Although I personally have moderate confidence in the correctness of the above hypothesis, according to Karl Popper's philosophy of science, a theory or hypothesis is scientific only if it is falsifiable. Therefore, to make my theory more compliant with scientific standards, I will now propose 2 testable predictions based on my theory for experimental verification. We previously hypothesized the counter potential profile of H5 horizontal cells, which contains exactly 16 activation intervals across the wavelength spectrum as shown in the figure. I further predict that the human retina should contain 8 grades of horizontal cells, with their wavelength spectrum activation interval counts corresponding to the values specified in this table. In view of this, my first testable prediction states, in the human retina, 8 distinct grades of horizontal cells should exist, with the activation interval counts across the wavelength spectrum precisely being 2, 4, 8, 16, 32, 64, 138, and 256 respectively. Furthermore, based on my conjecture regarding horizontal cells modulation mechanism, my second testable prediction states, within any synaptic terminals of any horizontal cell, there should exist a type of biomicromolecule functionally analogous to a J1 cycle component. If both of the above predictions are experimentally confirmed, it will mean my theory is most likely correct. If either prediction is experimentally falsified, it will indicate that my theory is either incorrect or at least requires revision. In my monograph exploring the human eye, I collectively refer to these 2 testable predictions as prediction 7. If you, the viewer, are currently affiliated with a biological laboratory, I strongly recommend sharing this video with the lab director and discussing this verification experiment with them. Based on my projections, should prediction 7 be successfully confirmed, the pioneering laboratory will gain substantial recognition. Their findings will likely be incorporated into standard biology and neuroscience textbooks, generating profound academic impact. This forecast stems from the fact that experimental confirmation of prediction 7 will empirically resolve the following four long-standing mysteries in neuroscience. Question 1. How does the biological nervous system convert creative potential signals into action potential signals? I.e., how is its analog to digital conversion achieved? Answer. The biological nervous system employs the Z1-circle method to accomplish rapid analog to digital conversion. Question 2. What functional role do horizontal cells play in the retina? And what is the working principle behind their function? Answer. The primary function of horizontal cells is to facilitate the analog to digital conversion mechanism in the retina. Their working principle involves applying counter potentials to bipolar cells during each of the bipolar cells should be resting intervals, thereby endowing bipolar cells with specific Z1-circle response characteristics. This provides bipolar cells and their downstream ganglion cells the functionality to encode a specific bit position. Question 3. How many colors can human eye exactly distinguish? Answer. If the human retina indeed contains 8 distinct grades of horizontal cells, it would mean the response values of photoreceptors in the red, green, and blue channels are each converted into 8-bit binary code. From an encoding perspective, the theoretical number of distinguishable colors would then be 2 to the 24th power, which equals 16,777,216. To take a step back, even if future evidence shows there are not 8 but n distinct grades of horizontal cells, the theoretical color discriminating capacity could be quantified by 2 to the 3nth power. That said, I maintain high confidence in my predictions of exactly 8 grades. For those wondering why I'm so definitive about this specific number, no more, no less, the detailed analysis is available in my Zhihu column. Question 4. What are the encoding principles of biological nervous systems based on spike chains? Among these 4 mysteries, this one holds the greatest significance, as it is one of the most pressing core questions in neuroscience. In the 2021 list of 125 scientific questions published by the journal Science, this problem ranks second among the 12 frontier challenges in neuroscience. If prediction 7 can be experimentally confirmed, it will provide the ultimate answer to the century-long debate over neurocoding principles. This is because, if validated, prediction 7 would demonstrate that the human retina encodes photoreceptor response amplitudes by on of arrays of 8 ganglion cells. Consequently, this will indicate that biological nervous systems do not employ temporal coding strategies, but rather synchronous coding strategy, where the fundamental information unit is the on of array of a neuron group within the same layer, with each frame of such arrays corresponding to a binary code sequence. As a side note, in my monograph exploring the human eye, I analyzed this very question, whether biological nervous systems use temporal or synchronous coding from a distinct perspective on encoding payload, and came to the same conclusion stated above. The detailed reasoning is available at the link below. Now, I believe you can appreciate why the lab that first confers prediction 7 will achieve lasting renown. Should prediction 7 be validated, the answers to these 4 mysteries will most probably be incorporated into core textbooks of biology and neuroscience, with the pioneering lab's name, experimental data, and conclusions recorded in those very pages. Of course, this work's potential inclusion in scientific history hinges on prediction 7 being correct. If it's ultimately falsified rather than confirmed, the lab's amount of effort may culminate merely in debunking obscure researchers' outlandish conjecture. This risk undeniably exists. Whether it's worth taking depends entirely on your judgment. This concludes our exploration of biological analog to digital conversion mechanisms. For those interested in further information, the relevant monograph chapters are available at the link below. Before we end this video, I'd like to reintroduce my visual neuroscience monograph, Exploring the Human Eye, Theoretical Hypotheses on Visual Neural Mechanisms. This book details my theoretical conjectures about mechanisms in human vision, including photoelectric encoding mechanism, analog to digital conversion mechanism, color information encoding mechanism, edge information encoding mechanism, motion information encoding mechanism, and stereoscopic vision mechanism. While I can't guarantee absolute correctness, I can assure you each topic has been thoroughly explored with depth that respects readers' time. Interested viewers can access the serialized content of this monograph through the link below. Again, here's the sharing declaration. This video and its materials are freely available for public use under the 3 specified conditions. You are welcome to download the original video via the link below and share it on other platforms or with the professors and fellow researchers. That's all for this video. Thank you for your watching. I'll see you in the next series.